Let's see, I think he's still trying to get in. Maybe I will. Let's see if he jumps in with the main. Just seeing if we send him just the regular registration, if that will somehow clean things up for him. So we're going to try to take him off as a panelist and see if he can jump in through the regular link. Thank you for your patience. Bear with us. Don't know why this is happening. It worked great for Professor Rawmakers last week. We're going to try deselecting him as a panelist. Delete, delete. And there he is. Hello.
Hey, am I in here? Yeah.
You're there. There's an image of you, but it's not your face. I mean, it's not video of you. So apologies for whatever has been happening from my end. I'm trying to see if I can promote to panelists. Let's see if that works.
Hopefully.
Fingers crossed.
Join us panel.
Okay. Says you'll be rejoining.
Hey.
Wow. Okay.
Okay.
Thanks for your persistence, and thank you everyone for your patience out there. Okay. Yay is right. Okay, we've got a nice side crowd here today. Let me go into welcome to REMAP Therapeutics, second webinar with Dr. Now correct me if I'm wrong, Manoj Das.
That's good. Yeah, Manoj. Yeah.
Noted researcher, Dr. Manoj Das, post-doctoral researcher, or has it since accelerated beyond then?
Yeah, no, I'm a super postdoc.
Super postdoc. I think that's what everybody wants to have on their CV. He's a noted researcher there at the Center for Psychedelics and Consciousness Research at Johns Hopkins, has been a lead author on numerous papers, including the one that brought us here today. Let me just do a quick screen share. I believe it's this one. And we'll do a shift to share a couple of them. But we're going to go for this one. Let's try that. Not that one, though that's a very interesting one. We're going to we're going to the other. Bear with me, folks. Here we are.
curious which one you're about to pull up.
The main one on the no, still that one.
Oh no, I think it was the other one, the one that you're talking about, the thalamic gating, where we talked
Yes,
about
the
different
thalamic
models.
gating one.
Yeah, that was the one you just pulled up a second ago.
Oh, okay. It was.
Yeah.
Okay. I'm sorry. It's showing me something different. Okay. Then in that case, so you and I got into an interesting discussion after Zeus Tapato. He put out a question like, who thinks there is I believe, a unifying theory of psychedelic activity within the central nervous system, or what is the best grand theory out there? So there were several answers. I put out like as much familiarity and kind of utility I've found with the Rebus model. I liked, based on what I've been doing through my work, because it's the one that I was first introduced to. I really enjoyed Gould-Dolen's critical period reopening via oxytocin induced, but then someone else chimed in, maybe it was Matt Wall, about not even the Rebus model, and you said, well, there's some issues there. And offered particularly in fact that thalamic gating as a model. Was it the striatal thalamocortical model?
Corticostriatal, thalamic, cortical.
Yes,
CSTC,
yes. So projections going back and forth, right? So
I mean, it's really kind of meant to be a circle, but yeah.
Right. So it's a network. So which instantly piqued my interest, because the thalamus is so implicit when we're talking about treating pain. And in fact, within the system I work at, it's like we have what's called an 8 levels model, and the thalamus is buried right in the center. And so it's receiving all this afferent information, and then it acts as sort of a kind of a warehouse, and yet a rechanneling, regating. A lot of people use the word filter.
Or relay. Yeah.
Relay is the word I was searching for. Thank you.
Yeah. So, I mean, that's actually one of our critiques of the model, or maybe more of an extension of the model, is that the thalamus isn't just a relay. And so there's this really interesting scaffolding. I think Murray Sherman's one of the big proponents of this. And essentially every projection to the cortex has a back projection back to the thalamus. Yes. And those back projections actually also branch to project onto motor regions. So this is known as an efference copy. So you get efferent signals that send from the brain to, let's say, motor areas, so leaving. Right. They exit. And so these kind of, the idea is that every signal that's being sent from the cortex, that's, let's say, like a motor command or something that's a primitive motor command, that is now getting also passed on to the thalamus. And so it's a copy of an efferent signal of a motor command. And with the idea being that now the thalamus it receives that information and sends us back to the cortex, but to a higher level now of the cortex. So now every low level motor command is being predicted by higher levels of cortex. And so, this, that's like just an extension of the model that I mean, it's not even an extension. I don't know how it fits in because it doesn't include necessarily the striatum. And I should say that I don't think that every psychedelic drug effect or, that the thalamic gating model alone probably accounts for everything. But I do think that just the data across different groups is just far more consistent with the thalamic gating model. I mean, essentially, the Rebus model is based on 2 brain imaging papers, and they don't even really go into behavior. They go into very kind of selective behaviors, like saying that in early visual behaviors or early visual processing, so for example, contrast detection is not really impacted by psychedelics, but higher level vision, so like motion processing, is impacted and it is impaired by psychedelics. But then they don't go into really high level, like types of, I guess, cognition, sort of like priming. So semantic priming is an even higher level you can imagine than motion perception. That's further along in the visual hierarchy. And in fact, that is something that semantic priming is something that I don't know if a lot of other animals necessarily have it. Because it's really about kind of meaning and these kind of high level concepts rather than the percepts. I'm sure a lot of animals do, and they have probably different things they can be primed for. But the point is that it's higher level than probably motion is, and yet that's been shown to at least be enhanced by psychedelics. So Rebus just kind of takes a lot of selective it takes selective behaviors, and then it also is based on basically just 2 imaging studies, whereas Robin's own data supports some of the thalamic gating model. So I don't know. That's just kind of, yeah.
You were just getting into a back and forth with him on Twitter about that.
Well,
I think over his new paper, and
yeah,
then,
that's a whole different issue.
No, I have seen that there is a lot of what shall we call it? People are getting a bit piqued around some of what's going on there. And it's like, it's at a level that is frankly far beyond where I'm at, since I know nothing about how to set up an fMRI study, what type of data is supposed to be, emphasized where, obviously trying to match all that. And there seems to be some discussions about, whether using the primary versus secondary measures from which study and things of that nature, arguments about using the kids SR versus the Beck depression inventory, which was obviously, I think, in silo dep 1 and silo dep 2, but in one it was primary and the other was not.
The quids was actually the primary in both of them. If you read the original papers, that's what was actually a little bit perplexing, maybe I should say, about why they said that the Beck's depression inventory was the primary measure in this new paper for the older data set. So from the original, was it Lancet Psychiatry, I think is when it was published in 2016, I think. So that said very, very clearly that the quids was the primary outcome in there. However, in their, they did have a clinical trial registration in which it said that the Beck's depression inventory was going to be the primary outcome. So
I remember
in one of these, and that's actually what they cite. They cite their clinical trial registration. They have the link to it as that being their primary outcome. So it's in one of these cases there was misreporting. Either in the original Lancet Psychiatry paper they misreported and said that the quids was the primary outcome, or in this paper there's a misreporting saying that the quids wasn't the primary outcome, the Beck's depression inventory was. And I mean, just as easily they could have looked at the HAM-D, which is what we used in our depression trial. And so it's just like, okay, just go with what the primary outcome is and what what you'd originally reported, which in the most recent sample it was the quids. And so I mean, and actually for that matter, by having larger variability in terms of efficacy, you might even be able to find better correlations. But my guess is that they didn't find that, which is why they were reporting on the Beck's depression inventory. Although, I don't want to make any accusations here.
Wasn't there also an issue though when they put it out through the New England Journal of Medicine, the Imperial College silo versus escitalopram study, that it seemed like there was fairly strong evidence that when they showed no statistical significance between the SSRI and silo, that it appeared to be a false negative based on what was going on with all the secondary measures?
So I would not say strong evidence. One of my colleagues, Sandeep Nayak, he just did a Bayesian analysis of it, and it seems like there might be some evidence, but I wouldn't say strong.
Okay.
So yeah, I mean, if I had to look at this, I would say, yeah, it does look like it would. If they had a larger sample, it does look like psilocybin outperformed citalopram. Also, what's a little weird is that citalopram worked after 1 week, which that just doesn't happen. If anything, things like suicidality goes up in the first week of taking antidepressants, but in the first couple weeks anyways. But yeah, so I mean,
I think that was a measure on that study too. That
Yeah,
they showed that it definitely went up for citalopram. That the suicidality.
Suicidality? Oh, really?
I'm pretty sure because I remember watching the BBC documentary about it, and they were noting that had in fact gone up, but suicidality definitely went down for psilocybin, which is just, full disclosure, something I personally experienced when I went through the study at NYU. Like, it dropped off the charts.
So that's interesting. I mean, one of the reasons I was going to say that the citalopram group showing a response at 1 week, which this is why it would have been nice to have a placebo group, is that, yeah, the massive doses of psychotherapy that you get, because they still got those, all the psychotherapy that you would get, that's standard now with psychedelic treatments. It just kind of goes to show that a lot of what we see with psychedelics has nothing to do with the actual drug itself, but rather it's about a lot of the like warm people being around and the hippie fuzziness that surrounds these drugs. As far as the decrease in suicidality goes, if you look at the Compass study, which a lot of people like to give grief to Compass, but in their press release they didn't have to say the number of serious adverse events
for the TR. Just so everyone's catching up here, this is the one that they released a few months ago. Was it September, I think? And if I'm correct, because I'm trying to remember when Wonder whatever was going on in Florida. I think for them it was like 240 participants in the TR treatment resistant depression study for psilocybin.
And I think it was like 6% in the 25 milligram group and 7% in the 10 milligram group that serious adverse events went up, whereas it was like less than 1% or something like that, or maybe 1% went up in the 1 milligram group, which that does probably suggest that after psilocybin treatment that suicidality can also go up, which, I mean, they don't say what the serious adverse events are, but I think they could include either suicidal ideation or self-harm or even attempted suicide, and hopefully that last one didn't actually happen. But
that's not unusual for that group too, right? So like if we're contrasting treatment resistant depression with major depressive disorder, right? Isn't suicidality a higher risk within the TRD arm?
I would imagine so. And I mean, it's also worth mentioning that I think in a lot of these cases with most antidepressant treatments, that they probably is in a risk of these things going up. So, I mean, one possibility is that people are just disappointed that something didn't work for them, right? So with SSRIs, for example, I mean, people get told that, hey, you're not going to get better for at least a month. Sometimes it even takes 2 months for these things to work. And yet they still probably have high expectations. And after a week, nothing's working. And then, suicidality might go up. I mean, the alternative, though, I think, is that people do think that certain things can still get activated, but mood doesn't actually improve. So things like maybe arousal or just general energy or whatever it is can happen after the first week or couple weeks of SSRI treatment. And there are subtle cognitive changes you can detect within a week of an SSRI treatment. And I think even within 1 day of an SNRI treatment, even when mood doesn't go up. So if that is going up and mood doesn't, then it might make people act on some of their I guess, maladaptive thoughts. And so, I mean, I think that this can be true across many treatments. So I know that I think with like bipolar disorder, like nearly every treatment that improves depression and bipolar can increase mania. So I think TMS, so non-invasive brain stimulation, definitely SSRIs. And my guess is probably psychedelics we'll see a little bit of that. So, I mean, there is a trade-off here, but I think, we always have to be aware of these things and not just slide them under the rug,
So just going back to two things you said. So in the case of the Imperial study prior to this, you thought that maybe just the environmental effect of everybody receiving extensive psychotherapy was in some ways more responsible for the outcome than the psilocybin itself?
More responsible is difficult to say.
Sorry.
Without a proper
Or maybe
placebo
a
group.
stronger contributor than we're giving it credit for.
Yeah, but I mean, if you look at, I'd have to go back to see those graphs to see exactly how
Sure.
big of a difference it is. I mean, actually, I want to say that I could be wrong here, but that the citalopram group did have a decrease in that first week. So when citalopram should not be working, that was over half of what the decrease was with psilocybin. And so if that's true, then yeah, that would be suggestive that most of the effect is coming from just, all the psychotherapy that they get.
There is
And it's not
also,
just
though, a question about, like, some people had not been adherent to the washout period, I think. So some people that were supposed to be off their citalopram or other similar classes of drugs were like, had not. And then they're also wondering, like, did some people, were they in withdrawal? And so now that they were back on SSRI, it kind of like eased the withdrawal symptoms from that happening. And so then that's why we saw kind of a bigger spike. So there's some
I mean,
suggestion.
that's definitely true. And I could imagine that, I mean, I don't know this to be true. This is massive speculation, but
Go for it.
if somebody were on an SSRI, although I forgot what their washout period was. Was it a month or was it?
I think it was 2 weeks.
2 months.
It was
Oh,
like
2 weeks.
It was not long enough.
Yeah.
From
okay.
what I recall. I mean, someone
So-
will correct me right away. Someone is putting something in the chat.
It looked like they put I saw the NEJM,
Right.
so the New England Journal of Medicine, I'm assuming it's the paper, but it's the, if you have, so somebody who's probably never taken an SSRI, or it's been a really long time since they have taken it. My guess is in people like that, it's going to take a month, 2 months for it to work, right? But in somebody who's only been off of it for 2 weeks after being on it for a really long time,
Yeah.
I can imagine that it probably works faster, in part because things haven't fully cleared, especially with 2 weeks. I mean, Prozac it takes a lot longer for Prozac to clear, for example. I don't know with citalopram, it might actually be a lot faster. But there's still these longer lasting changes. So for example, downregulation of the serotonin 2A receptor, I think in some people, just based on, this is all just anecdata, and this is something that, we really need to parse out better. But it seems that in a lot of people that the downregulation of the 2A receptors seems to be, if not permanent, like it does last, it persists beyond clearance of the SSRI. And so, I mean, I think that this was talking to Ben Kalmindi, and I think that this might have been at least somewhat true of in the anorexia study here, but don't quote me on that. That people, there was people who didn't have a large response, certain patient populations. So in Belkamindi's studies, it was OCD patients, and then over here it would be some of the anorexia patients. But maybe this was true in the depression trial too. I can't quite remember with everybody I've spoken to, but the people that seem to have a reduction in response to psilocybin, to these massive doses that we give people, and in some cases had like very little at all. Those were people who were on longer lasting, they were on SSRIs that had longer half-lives. So it seemed to be, again, there's no analysis here. This is all just based on, anecdotes. Sure. But it really seems that those SSRIs like Prozac, or fluoxetine, that they seem to probably have longer lasting impacts. Maybe it's due to the half-life, maybe not. In terms of, downregulating the serotonin 2A receptor. And then, there was that recent study by, I think it was Matthias Leichty's group, where they actually had people on an SSRI for 2 weeks, and they basically got no reduction at all, which does, it does suggest that, yeah, usually people are on SSRIs for a longer period of time than just 2 weeks, right? And, at least again, with anecdata, I can think of one person who I know who was on Prozac. And I think that the first time they did like psilocybin containing mushrooms, they had a really large effect. And they didn't even take a super high dose by any means. I think they took a pretty standard dose, but they were only on it for 6 months. I mean, compared to, which is still a long time.
I mean, that's compared to what you hear these days. I mean, so many of the people I think that often come to these studies, and just since I've gone through this, so many people want to know, like, the first question is like, can I take it with my SSRIs? The second question, I guess, is like, does this work for bipolar? And then I have to like give all the cautionary notes about like, well, right now that's an exclusionary criteria, though I know Josh Woolley is starting to investigate, so, but like the washout period, I think that they're asking for on a lot of these studies is too short for some folks. Though there's that recent, I think you mentioned the same author, the German one, didn't they do a recent study in this past year, maybe it was October, where they were like using, maybe it was in healthy normals. It was probably in healthy normals. SSRIs and psilocybin together, and show that there was
no reduction. Exactly. So it was 2 weeks, and I can't remember which SSRI. It was. I assume it was probably citalopram.
I think it was.
Yeah, and so they gave that for 2 weeks, and they didn't show really any reduction, it looked like. And so, as I said, I think it might be a longer period of time. And I don't know how long. As I said, the one person I know who's taken Prozac and took psilocybin-containing mushrooms, they were only on Prozac for 6 months. I think they even went off of it for a week afterwards, and then they took psilocybin, and they got they got a large response, from my understanding. So I wasn't there, but I definitely heard from them. And so I don't know. I mean, and I'm sure it varies between different individuals. There's I had 2 friends that were both on citalopram. Maybe they were on different doses. But the female had no response to psilocybin containing mushrooms for the first time. The male, on the other hand, had a normal response, I think. Again, I wasn't there, but I think that that was what they reported when they took them together. And so it just seems to vary. And I think both of them were on SSRIs for long periods of time, at least a few years or something.
I've heard that anecdotally, like people going to retreats that have been like years long users of SSRIs and other similar antidepressants, where they're taking like, 5 grams, and nothing. And so like some of them had to get worked up, considered like 3 times as high. And then they had a clinical response. But
I do worry about, how high we take these things. I mean, the fact is, we're also, I think, by we, I mean the people who are here working on the anorexia study. I'm not one of those people. I think a lot of the patients there might not always show a response, and so they might be taking the dose up. Although I think there's definitely a limit. But I mean, I do wonder, you start to worry about some of the other receptors. So there's downregulation of the serotonin 2 receptor. I assume there's probably with SSRIs and these things, I assume there's probably also a downregulation of some of the other receptors. But there does start to be issues with, for example, I mean, everybody talks about it, the serotonin 2B receptor and cardiotoxicity. So yeah, I do start, wondering. I don't know what to tell people who are on SSRIs. I mean, in general, I tell people don't take drugs, but
Are you aware though of any strong evidence in any cohorts out there that have been like taking, like say mushrooms or LSD, like just like, Grateful Dead or Fish fans, like, do they show like this evidence of like severe cardiotoxicity out there? You know what I mean? Because I hear people being worried about it because they know what 5-HT2B can lead to, but then we don't. I don't know if they're saying like, we're seeing this emerge in these, groups that have been doing this for a long time. I haven't heard that coming back.
There was, where was this? Was this from? This might have been on the Cover Story podcast. What is that called? Okay. Cover Story. I forgot the other. It's got like 2 names to it. Power Trip Cover Story. That one. Okay. So I think it was on there. Although maybe this was an article that was mentioning that, and so I'm just kind of blurring these 2 things together. But there was a somewhat older guy, maybe in his 60s or 70s, that they did report died from, I think it was just psilocybin containing mushrooms, and he did have a potential heart problem, I think. But that was like within an hour of having taken them, or was it like 24 hours or something like that? And like potentially that could have just been luck of the draw. Yeah, that this person just happened to have a bad heart. I mean, again, if somebody has a predisposition, any sort of heart problem, I would never tell them to take these drugs. I mean, then there was, I don't remember if it was Brian Roth's work, I want to say it was probably his, but let me not, again, don't quote me on that.
Sure.
But I think he showed that it's with the serotonin 2A agonist in animals that I think constant activation of the serotonin 2A receptor. And I can't remember if he used something like DOI or if he used something else. That there was evidence for cardiotoxicity. I mean, I think that's what
Okay.
everybody is citing, where that comes
Okay.
from. And I mean, the animal model is definitely applicable in that particular case, is almost certainly applicable to humans. So yeah, I mean, yeah, I believe that that's probably true, and I wouldn't sit around, microdosing every day, for example, because of that potential issue. With that said, I mean, there's rumors of, people like Albert Hoffman supposedly, microdosed until the end of his life. I have no idea if that's true or not.
Yeah.
Again, this is just things I've heard. And he lived to 100 and something, but I don't know, maybe I'm just more cautious.
Could we go back to you were briefly mentioning okay, if we go into your paper, which is awesome, and just full disclosure, like, so
Wait, which paper this time? The one
sorry,
that
sorry,
is the one
sorry,
that didn't get cited by the paper that was released this week, or the circuits paper?
no, the alternates to the Rebus model paper.
Oh, okay, yeah, the circuits paper, yeah.
Okay.
Okay.
The circuits paper, sorry. Just for people who aren't familiar with these structures, maybe as innately as you are, could you describe what the main actions of the striatum, like, so we've got the dorsal and frontal striatum, and then you also have the thalamus. Would you mind just briefly describing what's going on there and why you think that they deserve more of a focus? I mean, I also recall reading in your paper, you talking about how this has an effect as well on the ventral and dorsal visual fields, and then that they're getting down into, like, I think you said the same, somehow there was an association with the layer 5 pyramidal neurons in the frontal cortex. I'm just curious.
Yeah. Okay. So, I mean, first of all, one of the things we do mention in there is that there's probably more going on than just the layer 5 pyramidal neurons. Wait, I just saw something in the chat. Somebody said something about Brian Roth. Brian work.
Brian Roth.
Stimulative. Yeah. Brian Roth. Yeah, there you go. Okay, so it was Brian Roth. Great thing that I'm glad I attributed that to him. He's a great scientist. So there's potentially more going on than just the layer 5 pyramidal neurons. That's the one. Those are the ones that everybody talks about, and it seems to be that's where serotonin 2A receptors are. Most of them are. And so these neurons are excitatory. So when you activate them, they will send a action potential that's now going to potentially activate the next neuron.
Right.
And so, but there are other serotonin 2A receptors that are distributed not in the cortex. So those are in the cortex, right? So layer 5 is in the cortex, but there are other ones. For example, there are serotonin 2A receptors in the actual striatum, in the thalamus, some in the hippocampus, also in entorhinal cortex. So, and we talk a little bit about the ones in entorhinal cortex because that's the entrance to the hippocampus, and they're actually on inhibitory neurons there. And so, maybe more on that later. But as far as the ones that are in the cortex go, at least, yeah, some of them in the original in the original thalamic gating model, they talk a lot about the medial prefrontal cortex and or the dorsal anterior cingulate. And the idea there is that in these layer 5 pyramidal neurons, they project now to the striatum. And so these are subcortical structures composed of like the caudate and putamen. And what's interesting about those is that those are inhibitory. So they send inhibitory projections to the globus pallidus, which is also now part of the basal ganglia. So those three structures are part of the basal ganglia, the caudate, putamen, and globus pallidus. So now you're getting the striatum, which is essentially the caudate and the putamen, and then they're sending inhibitory projections to the globus pallidus. The globus pallidus is sending inhibitory projections now to the thalamus. So what that ends up being is that you get essentially two negatives become a positive. So by exciting the first set of inhibitory neurons, they're now going to inhibit the inhibition of the thalamus, because the globus pallidus is inhibiting the thalamus. It makes a lot more sense if you look at the graph, but essentially you're inhibiting the inhibitors which allows now the thalamus to now send whatever it wants out into the cortex. And so some people, unfortunately refer to this as like, oh, it's like the filtering mechanism has gone down. And there is filtering that's gone down. But it's not like, the thalamus is filtering things that we can't normally see. It's more like an attention impairment. So for example, if I'm sitting at a cocktail party, and I say that because this is known as the cocktail party or effect, I think. But like if I'm at a party, I can focus in on what you're saying, even though there's all this noise going on in the background. At the same time, I can also, not really focus on what you're saying and try to pay attention to what the dudes next to me are talking about, right? And so in contrast, under psychedelics, now that filtering can't really happen as well, that my ability to filter using the thalamus attention, that it's now just everything's going to kind of come in. And so because of that, they're not coming in well, because now I can't focus on anything. So, people like to think of this as being like a good thing. And it's relative to what a good thing is, but yeah, this isn't essentially here it can result in impairments in attention, for example, and a lot of I guess there's other potential explanations of certain psychedelic phenomena like color constancy that might also be related to this. So color constancy is seeing, for example, I can look at this wall and say that it's white, it's a white wall and all along the wall is white, when really if I were to take individual pixels out, blow them up onto a white screen on a computer, I would see that there's probably a full range of colors, including probably some reds and some grays and whatever else.
Well, the thalamus is responsible for sensory discrimination, correct? So like the ability to sort out different sounds. So like if you're doing something like tonal topic mapping, you should be able to hear like, okay, this is one pitch, that is another, this is coming from this type of object, this sound is coming from that type of object. And like, I think it's implicated in things like mirror box therapy, when we're talking about, treating people with certain types of pain. Often they have like very poor sensory discrimination. And so initially, like if you're doing like implicit motor imagery for mirror box therapy, you have to start off with stuff where it's like, okay, this is just a frozen picture of a left foot or a right foot, which is it? And then kind of as people are working through, this is called graded motor imagery for working up to mirror box therapy, then they start going to like, so from say, third person association to first person association. So, and you're slowly working up through imagery that's like going from kind of isometric pictures to ones that are actually in movement there, and that are personally associated. So that kind of ups the level of sensory gating that's going on. But from what I understand, the thalamus is responsible for sort of fine discrimination.
I
Right?
don't know for certain. I mean, look, I'm not a thalamus specialist, but at least a lot of these like really fine-grained discrimination type things, you might expect that more in the cortex. But I don't know. Maybe I'm wrong there. Maybe I'm just being corticocentric right now. I think none of these things are isolated necessarily to one area, and it's always, and it's about being a network. And especially what I just told, what we started this conversation with is that everything in the cortex projects back to the thalamus.
Right.
So
and there's like 30 nuclei in the thalamus, right? I mean, it's like it's a massive hub in the center there that like everything is going back and forth.
Yeah, and I mean, it probably also matters on which nuclei you're talking about. So at least, for example, in the hippocampus, fine-grained resolution of memories or even perception is involved in more of the dorsal regions or the posterior regions of the hippocampus. And as you move up, things become like the resolution is kind of lower, although it becomes more generalized and useful for like extracting regularities and forming schemas. But yeah, I mean, I would imagine that could be the case too, because early processing of visual information is going to be probably the most precise you get, and then as you then project further up, it's going to take more kind of taking the general picture. And so, yeah, I mean, like, for example, if you take the cortex, the early visual cortex, you can, a lot of times you can basically see what the person is seeing, not literally. I don't think you can fully decode an image from visual cortex, but that would be your best place is V1. And then it gets a little murkier as you move up. But yeah, I mean, I don't know about the fine-grained discrimination exactly in the thalamus. I mean, you could just, especially, I mean, a lot of these decisions and things that reach like consciousness, let's say, a lot of times involve further up areas. And so, you know, V1, for example, early visual regions, you're not really necessarily conscious there. It's only whenever you get frontal like interactions with what's going on back there. I mean, this is if you buy at least certain models like Akwin-Lau's models, higher order consciousness models. Yeah, then you start to get, you start to get more access to things that you're conscious of. But I mean, I guess you can still make fine-grained discriminations without actually being conscious of it. I mean, that's sort of like what is it, blind sight, for example, where people don't, they say they can't see anything and yet they can still perform above chance.
Right.
So, I mean, yeah, I don't know about these fine-grained discriminations, but I guess, yeah. One thing I guess I wanted to say was you asked earlier about why I think, what the data go with it. And so at this point, there was Robin's study with the psilocybin data that were reanalyzed along with the LSD data. I think the study was in Current Biology, and Enzo Tagliazucchi was the first author. And then there's
and they were chiming in today on your thread back and forth between you and Robin, I think.
Yeah, it's probably, I'm tempted to talk about that right now, but
I mean, if you want to, I mean, it's
yeah,
an open forum. I mean, I would probably then ask Robin to chime back in like on another episode or something like that, but up to you.
Yeah, I mean, I'm hesitant because let's just say we did come up with something very quickly in response to their paper, but yeah, so I mean, just to go back to the thalamus stuff real quick, if there's time, we can talk about that.
And also,
paper.
yeah, we got started late, but I mean, we should give people a chance to do a Q&A as well. But
oh yeah,
please.
Yeah, and maybe it'll come up there. Yeah, I'll save the paper that came out this week, which is completely irrelevant to what you wanted to talk about. I'll save that for if anybody wants to ask in the Q&A. But yeah, so with the thalamus. So in their paper, they looked at what's like known as global functional connectivity. It's essentially taking the average functional connectivity of a region with every other region. So the functional connectivity is how two regions in the brain, how their activity kind of co-varies over time. So for example, two regions activity that are doing this would be highly functionally connected. Two regions that are kind of just doing whatever they want, it wouldn't be assumed that they're necessarily talking to each other, at least definitely not directly. And there's probably not even a third region mediating. It would be further probably up in number of connections. And so the idea there was to look at how connected is one region compared, with everything else. And what they found that was, interesting was, oh, hey, that global functional connectivity increases in these higher level cortical regions, like regions of the default mode network, salience network, et cetera, but also in the thalamus. And then Katrin Preller came out with a study in eLife under LSD also. And oddly enough, she showed the opposite, that global functional connectivity, she did do things slightly differently. But we can talk about what she did at a different time, but she did show that global functional connectivity increased, but in sensory areas. So not in these multimodal association cortices, higher level cortices, whatever you want to call them, but in sensory areas, but also the thalamus. So, there was slight differences in methodology. One of them was that Enzo took full regions and then looked at the full regions connectivity with all of the regions. Katrin, on the other hand, took literally every single voxel and took its functional connectivity with all of the regions. You wouldn't imagine that should change things too much, although it's not impossible that could. The other thing that Katrin did is known as global signal regression, and that's where she got a lot of grief from Robin. And I found out that he emailed her and saying we shouldn't disagree. And then in a really funny thing, she later published a paper with psilocybin where she looked at global functional connectivity and completely, even though she also used a receptor map, she completely did not cite their Current Biology paper, which also used a receptor map. They both used two different receptor maps, though. One was based on PET imaging data. One is based on, I think, gene expression from people who died, I think. And so the funny thing is they both said, hey, look, our gene expression maps map on to the areas where we found increase in global functional connectivity. But the one thing that was consistent across their studies was the thalamus's connectivity increasing. And then for that matter, Matthias Leichty, so in Basel, we've now mentioned him. I think he was, I'm pretty sure he was an author on that. I don't think Franz's group at all was authors on these papers, but they also showed that functional connectivity in the thalamus, global functional connectivity, as well as they used a different method in a second paper. They showed that its connectivity increased with the cortex. So that is the most, evidence that there is so far that is consistent. There was also some studies showing that there were changes in thalamic activity with PET imaging, although it's not always consistent. And if I'm not mistaken, I think Robin showed in the initial psilocybin fMRI paper that I think that there was, if the thalamus was there at all, that there was decreases in activity if it came out. And so, I mean, I think that there's a couple issues here, and we mentioned this in the circuits paper. And one is that you're letting people do whatever the hell they want in the scanner. That is already a flaw in these designs. Resting state is the lazy man's version of scanning people, and that's coming from somebody who uses resting state sometimes. So I think that, yeah, we need to move away from that and constrain cognition, so that way we know the cognitive operation is being performed, because it could be the case that some sample of people, these are small samples, right? You're talking about 15 subjects a lot of time. I mean, Catherine's study and the groups in Switzerland, they're running more people usually. But yeah, when you're looking at 15 people, if let's say 7 of those people are thinking about their mother, they just so happen to be, or doing something like that, autobiographical memory, that's going to massively bias the data. Whereas in another 7 people, they're, let's say, less experienced psilocybin users, and then they become paranoid that the researcher is trying to, try to read their minds, which we can't do with fMRI. Or alternatively, it could be some people who are just, really enjoying themselves and paying attention to the wonky sounds in the scanner. I mean, these are all going to produce different patterns of activity. And I mean, this idea that there's intrinsic connectivity is true, but it should be robust to whatever task they're performing. And we should at best try to constrain everybody's cognitive operations.
So just so I make that point clear, you're saying that when people go into an fMRI scanner and then they're, induced into a psychedelic state through psilocybin, at a certain point in that you give them a very specific task to follow, so that that's a control measure for all the people going through versus having all this variability. I mean,
exactly. And especially when you're comparing, because all these comparisons are being made, right, to placebo. Now, under placebo, there's one study that showed 40% of people in a non-drug study fell asleep in the scanner. And that was healthy people as well as people with mood disorders. So, look, I fall asleep in the scanner. I stopped doing my friend's studies a long time ago if they're over 45 minutes. And so I pass out in the scanner really hard. In fact, I participated in a sleep study once because of that in the scanner. And so it is true that people fall asleep, especially when we tell them to close their eyes. This was actually something that a bunch of us were on this paper together. Drummond McCulloch is the first author. He got everybody together, opinions on trying to come to a consensus on what we should do with brain imaging data and psychedelics. I was, 1, against the idea of resting state, and 2, I was against the idea of keeping people's eyes closed, because then you're more likely to fall asleep. If anything, one thing that we would do in my first fMRI paper, I'm next to last author. I tell everybody I'm second senior author, but really I was just the least important person on it. And this is my most cited paper. But in that paper, there was a resting state component, but we had people actually keep their eyes open, and there was an eye tracker that we could actually watch people's eyes to make sure they didn't fall asleep. And we were actually looking for something very specific in that rest period. We weren't just randomly looking at rest. We were looking for reactivations of something that they'd seen previously. And so anyways, there was I think we have to, so if people are falling asleep in the scanner under placebo, but we know that psychedelics prevent people from falling asleep, then what are you comparing here? You're comparing awake versus sleep. You're not comparing psychedelics versus not psychedelics. And so, Danny Bassett, she's done this once. She did a study actually with dextromethorphan, and they had people perform a working memory task. And conveniently, people didn't get worse on the working memory task with dextromethorphan. And so that in general, people were performing the same cognitive operations. They weren't necessarily just zoning out and just not performing the task. And yeah, then she was able to show, I think that made a lot more sense to then be able to say, hey, look, the brain becomes more flexible, and flexible here really meant not necessarily a good thing, because they compared this dextromethorphan group to people with schizophrenia and showed that, hey, look, a similar sort of effect happened, as well as first relatives of patients with schizophrenia. So That I think would be a better route. I also think just not just mass speculation on intrinsic connectivity and what it means would also be, this is known as reverse inference.
Okay.
So this is essentially abductive reasoning. So you can, conditional, you can say that X given Y, but that doesn't necessarily mean Y given X. And so here you can say that, yeah, when people, for example, see your fearful face, that your amygdala activity goes up. You can't say that the amygdala activity goes up and therefore people are scared, right? And so that is known as a reverse inference, and the amygdala's activation goes up for all kinds of things, including not even necessarily emotional things, just salience in general. I've said this before, and I keep waiting for a vision scientist to say this isn't true or it is true. But my guess is if you were to compare a checkerboard to a gray screen, you would get probably greater visual activity because there's more salience in a checkerboard than in a gray screen.
Wow.
But I could be wrong there.
Speaking, speaking of visual issues, so just to segue here into some Q&A.
Okay.
Zeus, I don't know if you can see this in your chat or not, but Zeus has, hey Manoj, thalamic gating is such an interesting theory to explain the subjective effects of psychedelics. Personally, I think there's more meat to the idea than entropic brain theory in regards to visuals. We know there's expanded functional connectivity with V1, among other things, but in regards to thalamic gating and visuals of a trip, there undoubtedly has to be some gating with the lateral geniculate nucleus. Like 80% of excitatory synapses found in LGN come back from the visual cortex. What do you think is going on there in regards to psychedelics? Giant question, I know.
Yeah, I don't know. I don't know if I can answer that because I also don't think that it's just about sensory gating. So for example, the best research in animals, as well as to some degree now in humans, is that the thalamus is not a monolithic structure, right? It's got a bunch of different nuclei, as you mentioned. And this is actually something that we kind of take issue with in the paper. In Rebus, they kind of refer to things, what's higher level and lower level is not always clear. And so, for example, it does make sense that early visual processing, V1 versus V3, yeah, V1's lower order. But then what they also kind of refer to as lower order is things that are in the limbic system, which I'm not really a fan of that term. Yeah, that for example, so the thalamus or the hippocampus would be considered lower level, as well as the parahippocampal gyrus somehow, even though the parahippocampal gyrus is essentially the top of the visual hierarchy. The dorsal stream ends at basically the parahippocampal cortex, and then the ventral stream ends at perirhinal cortex. And so,
just so people understand, ventral stream is generally for object identification. Dorsal stream is where is it in the environment. Those are those two streams that are being processed.
Where and what,
Where and what?
Yeah. Yes. And so, yeah, you end up. So these aren't though more like primitive regions by any means. They co-evolved with the cortex. And so, for example, in the thalamus, it might not even be so important that the visual projections are as important, but rather, for example, the medial dorsal thalamus. This is a region that projects to medial prefrontal cortex. I don't think it receives any primary sensory information. And that particular region that projects now back to, let's say, medial prefrontal cortex ends up being, there's a lot of work there showing that region seems to be somewhat important, especially with regards to the serotonin 2A receptors.
And the medial prefrontal cortex is part of the default mode network, which is cited is part of the Rebus model, correct?
Yeah, so the default mode network narrative, I don't know if I want to get into that again, but yeah, the default mode network narrative is, I mean, look, Robin himself, I always use this in my talks now. I always use a screenshot of this tweet that said, there's been an overemphasis on the default mode network narrative. And I'm like, yeah, because you double down on it in Rebus, even though his own data show there's larger effects. Here's the kicker right here. In the LSD data set. So, okay, let's go back to Rebus. Rebus has this idea that lower order regions are less affected by psychedelics. Lower order here meaning like lower level sensory areas versus multimodal association cortices, which I'll agree there that, yeah, that's a lower level in the hierarchy is like V1 versus, let's say, medial prefrontal cortex. I don't think anybody's.
Can you just rapidly describe for people what V1 is, since we've referenced it?
Oh, yeah, V1 is the first stage of visual processing after the thalamus. So back of your brain, occipital lobe. So, visual information gets projected to the thalamus. And then that now goes to now.
And then once again, the thalamus is a relay station that projects out into the cortex, which in turn goes back, sends efferent signals back to the thalamus.
Yeah, and so
that, are you sure you really want to stop subscribing? Or maybe I could just, instead of an email every day, how about once a week? So it's kind of in a discussion like that.
So and actually to go back to the lateral geniculate nucleus is the first kind of stop of visual information. There are, I think, there's blue cones that also project to suprachiasmatic nucleus, but let's forget about those for a second. But I think that, so most visual information will now get first hit the lateral geniculate nucleus. But, I'm forgetting now, but we wrote about certain thalamic nuclei in the paper. This was, by the way, a massive effort. This was supposed to be a paper that we were supposed to write within a couple months, and then it turned into like a year. And I think that's the way reviews should work, which is why it got into a decent journalist, because it took a bunch of different minds coming together, animal researchers as well as us, anatomists, et cetera. And so there's even an MD on there, and so I don't know if the LGN actually has serotonin 2A receptors there. I think that, or else we would have mentioned it, and maybe we did mention it, but for sure, there could be back projections from V1. So just to go back to what I was saying about the default mode network here, there's a lot of the thing about rebus is that the idea is that higher level cortical areas, the multimodal association cortices, those should be where the majority of action's happening with psychedelics. That is where there's a higher density of serotonin 2A receptors, although there's still a really high density of serotonin 2A receptors in cortical regions, at least compared to some of the subcortical areas. And so one of the issues with the LSD paper, not issues, is this is what the data show, and this is also what everyone else's data show, is that the largest effects are actually in sensory areas. This is true whether you're looking at functional connectivity. This was true in their entropy paper. In fact, in the entropy paper, the smallest effect in entropy was in the default mode network and the hippocampus. But the hippocampus, you get more signal there with fMRI, so we can forget about that for a second. But the smallest effect was actually in the default mode network, and the largest effect was in like somatosensory or visual areas. And so this completely goes against rebus, and yet they doubled down on the default mode network narrative in rebus when the data didn't even speak to that. So that was one issue with the default mode network narrative. And of course, I sound like a parrot now if you probably have seen anything else I've either said in talks or written about. But yeah, I mean, every drug seems to decrease default mode network connectivity, and I think one reason for that is that the default mode network is a network that seems to come on more when attention's not being paid as much to the outside world. And so if you're on a drug, you're probably more likely to be kind of shifting your attention back and forth between internal and external,
so does that mean that there's an increased interaction under psychedelics with interoception and maybe the insular cortex or something like that?
I don't know about that. I would not say that necessarily. I would say decreases in default mode network
questions when other people are waiting.
Oh yeah, no, I would say the decreases in default mode network connectivity could mean that people are just paying more attention to the outside world at times compared to when you're on placebo and you probably do go inwards, and you go so inwards that you fall asleep. So I think this is something that, if you're on drugs, so this shows up even with alcohol. And my guess is, you get drunk in the scanner and you're probably like having a little bit more fun, especially if you're on the earlier end of drinking, right? On the tail ends, it's probably going to be more sedating. But this shows up now with amphetamine. Amphetamine, for sure, you're going to be more awake and probably shifting your attention back and forth between what's going on inwards and outwards. Anything that's you're paying attention to the outward world, you're going to get less default mode network activity. And so this is going to be, this is just this is a confound with not constraining cognition in the scanner. And so again, every drug seems to decrease default mode network connectivity. I should say the drugs that are most similar to psychedelics that are not of the serotonergic, psychedelic class would be ketamine, the dissociatives. And there's, I think, 4 studies with ketamine now with resting state connectivity, 4 or 5. 2 of them show decreases in default mode network connectivity, 2 show no effect, 1 shows an increase. And those are all better studies than all the psychedelics. The psychedelic meaning serotonergic psychedelics here, in that they had larger samples. I think they better controlled for motion. One of them was multi-site, so it's a lot more generalizable. And they're not showing the same effects across all of them, which really tells you this is probably a problem with letting people do whatever the hell they want to do in the scanner. Okay. So again, yeah, this default mode network. This is one of the things we some of the issues we raise with Rebus compared to the thalamic gating model. But I mean, we do say that there is some potential things in there that might be interesting, especially this idea of something being, they explicitly make a prediction in Rebus about there being more effective connectivity, so more kind of information flow from the hippocampus out to the cortex. And they actually group in the hippocampus with the parahippocampal gyrus, which is not the same thing. And the parahippocampal gyrus isn't a monolith either. There's 3 different, like, very separate in terms of their function cortices: parahippocampal cortex, perirhinal cortex, and entorhinal cortex. And anyways, they actually showed in their own data that they get projections, more information flow from parahippocampal gyrus to the visual cortex, which is opposite what they state. But in any case, there is something potentially interesting there, I think, that involves probably gating into the hippocampus in those entorhinal serotonin 2A receptors that I'd mentioned at the beginning of all this. Right. So, yeah, that was I don't know what I think I just rambled.
No. Yeah. This has been incredible, honestly. It's just like just seeing that the volume of information out there to interpret is it's overwhelming, and it's good to know that there are other viewpoints and constraints being followed here. Mind you,
the volume of information that is out there in human brain imaging is vast majority based on 2 studies. So just keep that in mind from the same group. Yeah. So none of those, I think a lot of people don't realize that they're just 2 data sets that have now been reanalyzed such that there are more papers than there were participants. So it's not even very large, data sets. So this is why one group is kind of running the narrative, to say the least. And so, I'm not saying that. I think that they do great work at times. I think this week's paper, I don't think it was bad per se. I just don't think it belonged in Nature Medicine. And there were some issues with it that, you'll see sooner than later about, when this paper comes out.
Okay. I mean, I saw right away there was friction. And so I figured, like, and it was just fascinating that this happened. Was it yesterday? And like we were planning on doing this. And so I was like, when the paper came out, and so or was it 2 days ago? Sorry, I've lost track. But just the fact that we were already going to be discussing alternate models to Rebus, your critiques of it, and then here comes their fMRI study, specifically, and then you guys had immediate feedback on how you felt that one as well. So it's just it's a fascinating dynamic.
Yeah. So you can look at Twitter and just see what we had to say, but. Right. Yeah, I think the thing that, the first thing that bothered me when I was reading it. So Nature Medicine, first of all, is a really high impact journal. It's this is where like the latest like MDMA clinical trial went to, that had, like nearly 100 people or something in it. And what goes in there a lot of times will dictate what happens in the future. And I think that's for good reason a lot of times. A lot of really good work does belong in good journals, and then those good journals, will help influence what happens next. And so one of the issues here was that one of the findings, especially one of the findings they show, where they're looking at, it's not just functional connectivity across the whole time series, but rather you're looking at functional connectivity, how it changes over time. So 2 regions, for example, might be in sync at one point and then go out of sync and go in sync. And so a single functional connectivity value wouldn't be great. You need to estimate some time series of functional connectivity values. So that's something that I did in a paper that came out 5 months ago. And then I looked at how the variability of functional connectivity can change. And that's not too different from what they did. And they don't cite our paper at all. And the funny thing is they speculate that it might have something to do with cognitive flexibility. And we actually looked at cognitive flexibility, but this is a group that is known for. They're no, God damn it, Cat. They're known for not using behavior and instead speculating using Freudian models, which fuck Freud. Just straight up, I'm going to say that right now. I can't believe that he's penetrated psychedelic research. Like in most of psychology, he's now a joke, and yet now we're the joke of psychology research. So they instead will use Freudian like speculation on resting state brains instead of actually collecting proper behavior to be able to constrain the search space for what's going on in the mind at that time. And so, you can speculate on cognitive flexibility all you want, but you should have measured it. And that's what we did. And yeah, our paper had a bunch of flaws. I'll say that their paper for sure had, a larger subject sample. They had, essentially 2 time points. They had 3 weeks and 1 day after psilocybin therapy. And so, I mean, in that way, I think that, yeah, their paper was better than ours, other than they didn't have any proper behavior to show for it. And so when you're speculating about that, how are you going to not cite my paper? And, they claim that they didn't read it, but I was like, damn, 9 authors and none of them are keeping up with the psychedelic literature. Like, that just seems, and here's something, I'm going to go off a little bit, but we got a review from somebody that said something very similar to what Robin responded to on Twitter. And then for that matter, they also basically asked us to cite one of Robin's papers that had nothing to do with the paper, which we didn't cite them, by the way. Yeah. In that paper, we cite the things that we should cite of theirs, but in that particular case, it was like, well, no, this doesn't make any sense to cite. So that seemed like it must have come from somebody from Imperial, but I could be totally wrong, and I don't want to make any accusations here. But yeah, I think just the fact that, I mean, okay, fine, if you didn't read it, then that's not great scholarship in terms of keeping up with what's going on in the literature, So that bothered me, but there was a lot of other glaring issues, like poor statistics, 2 sentences in a row. There was, the potential of regression to the mean, which essentially means that if you already start, let's say you're an outlier, if your group happens to have a lot of outliers in it, and then that they, or if you're just a single outlier, you're likely to then go towards the mean the next time around, just based on, just general statistic. Look, you happen to perform badly, it was a noisy measure, whatever, next time you're going to be closer to the average of what everybody is. And so that's essentially what happened with their citalopram group, where they started, I mean, they weren't quite at floor, but they started at the same level as what the people who got psilocybin therapy went down to. So there wasn't much room for the citalopram group to go any lower. And so you can actually see that, yeah, people kind of just stay the same. And then where the psilocybin group starts high, they go down. So, I mean, there's some other issues I think we go into in this reporting, but to this reply. But yeah, those are some of the main problems there. I mean, I guess I've pretty much covered all the main problems here at this point. Yeah. So yeah, that paper, yeah, definitely rubbed me wrong. But I trust Robin's not a bad person. I used to live in London. I had drinks with him in the past. He seemed like a great guy. But yeah, I think that definitely rubbed us the wrong way.
No, that came off like immediately. So, and then it's like, I figured it's like, folks aren't just being petty out there. I mean, we know how rigorous like everyone is, trying to approach this, and then though I guess you guys are saying not rigorous enough on some level, or there's real.
Yeah, I think that in our paper too, we admit all the flaws in our paper too. And I don't know if they do that as well in their own paper. And like one of the things they mentioned, they talk about like rigorous motion correction. And I was like, wait a minute, you used a standard pre-processing pipeline for motion correction. I mean, at least nothing different that we do, other than we do more rigorous motion correction. There's a whole thing where you can use more motion regressors, and that's not something that they did. They use 6 when now the standard is really becoming 24. And in fact, that sometimes will help your data too. It helped our Salvia paper because Enzo Tagliazuki asked us, hey, what was the motion like? And it turns out he was damn right that there was a lot of motion, and when we corrected for it, it actually made the stats better. So yeah, there's just, it just seemed like that paper was a little dishonest. And I mean, you can just read Phil Corlett. He goes off on it, and he's somebody that we together sort of spoke to, and very quickly came up the fastest I've ever written anything. I'm not the fastest writer in the world. And I'm also not somebody that likes to write things super quickly because it turns out garbage a lot of times, as you now see in a lot of these psychedelic opinion pieces and reviews and whatnot.
Okay.
More people need to sit on their thoughts before just spewing garbage.
Oh. Well, I can't thank you enough for coming here today and trying to like bring some light to like other models and like just what goes into creating those and everything. Kind of the overall status of research as we're seeing it right now, and a little bit of pulling back the curtain on the process.
Oh, hey, real quick, can I?
Yeah.
It looks like Antonio and Sarah. I'm assuming this is a person. He had a paper in Pharmacological Reviews. I think so. That was on plasticity and psychedelics. So I kind of wanted to answer this question.
Of course.
Assuming this is the same person, I think it is. The reticular thalamus, the reticular nucleus of thalamus. He asked out. Yes, this is him. Okay, great. I don't know. I've never met this person before. But,
I was going to ask you earlier about the reticular nuclei that you were mentioning in your paper as well.
Yeah. So I think it's one of the regions actually that doesn't have serotonin 2A receptors. I could be wrong when I say that. But it does seem to be kind of like people refer to it like a pacemaker. And so some of the changes so if there's one consistency in resting state work with, in this case, electrophysiology, it seems to be the decreases in alpha power. Those have been shown, I think, back since like the 1950s with mescaline. And now across different groups, across with ayahuasca, with multiple ayahuasca studies, with DMT, with intravenous DMT, with psilocybin, LSD, et cetera. And so the reticular nucleus, I mean, it is kind of, I think it's more responsible for maybe even slower oscillations, especially for things like during sleep. But that could have something to do with that. We're not, it seems to modulate what else is happening in the thalamus. And so, yeah, I mean, I'm not super, clear on what's going on there, but I do think it's probably important for yeah, that's the best I can answer your question. Sorry, Antonio.
Okay. One new message.
Yeah.
No,
that was
okay.
just
all right. Well, let's we've gone an hour here. Just in closing, is there anything you'd like to add or clarify or emphasize?
Yeah, I mean, I think yeah, that we shouldn't always be quick to just pick up on a narrative, especially a narrative that a lot of times just repackages, things that the hippies have been saying, but using slightly, more scientific jargon. And don't believe anything I have to say either. I probably spew, in 10 years, we'll find out that I'm spewing a bunch of bullshit too. But I think that there's, it's worthwhile to try to find, I've learned nothing new about the mind from psychedelics since the psychedelic renaissance. And I really implore anybody to tell me what they've learned about the mind from psychedelics since this, the resurgence of this research. I've learned nothing about the mind, and I've learned very little even about their drug effects. In terms of what they do to the brain and with fMRI, kind of, but even then, I can't even say under what circumstances do we see these changes in activity and what those changes in activity even mean. Let's pretend it is true that consistently the deep follow network has reduced its connectivity. It also seems to be sensory areas reduce their connectivity. What does that even mean in terms of behavior? Or in terms of what's going on in the mind? I have no idea. The post hoc correlations aren't good enough. With random subjective measures, you literally have, hundreds of brain measures and hundreds of subjective measures. You're going to find something significant that correlates, and they never replicate. The replication, the ego dissolution, decrease in deep follow network connectivity and ego dissolution, that was found once with LSD. The Swiss Basel group, they did a better study with less motion and more subjects. They found a correlation that was basically zero with ego dissolution and the decrease in deep follow network connectivity. So I think that we just really have to be critical of what are we learning about psychedelics here. And I think that some of the times we don't need to learn anything. We can just say that, hey, look, we need to prove that they work for something like depression, and that's fine. But at least
what do you think then is going on with folks that are going into total remission? I mean, I'll just say, like, I was in total remission after 8 hours. I know someone else who went through the same single dose study I went through. 26 years of treatment-resistant depression since onset at adolescence, and they had tried all standard of care practices, and you can imagine an awful lot of alternative ones too. And finally, they did this, and like, end of the day, total
Right.
remission, and it's
Look,
sustained for a year.
it's clear that psychedelic researchers have either done the drugs themselves and gone through that process, or they know people who have, right? We wouldn't be studying these things if we didn't think there was something remarkable about them. And, you know, every psychedelic researcher other than one person has done the drugs. But, you know, yeah, the fact that I don't know, it doesn't matter. What I was trying to get at basically there is that sometimes it doesn't matter what these drugs are doing, right? As long as they work. But a lot of times what we want to do is, first of all, find out how to maximize the benefits while minimizing the harms, because these drugs definitely have harms,
Yes.
just like any treatment, you know.
Yes.
And that, you know, at least for those of us, some of us are, you know, actually this was a comment I saw Zeus had said, you know, I'm just interested in how these drugs, you know, work, and just like the random phenomena that come with them, right? I think that's somewhere where I'm at. I'm a basic scientist by training. I'm a cognitive neuroscientist by training, even just a cognitive psychologist. And, you know, I did want my work to have more impact in the world and to actually make a difference, because just pure memory research, what I was doing before, it wasn't helping anybody other than other memory researchers. And it probably wasn't going to have any impact in the clinic, for not in my lifetime anyways, even if it did have some effect later. So, you know, but I'm curious still, just at the basic effects of these drugs. That's just where my, you know, general training was, and maybe that's selfish. I still want to make sure that they are also helping people, but you know, these ideas that we're really learning how they're working, I'm just unclear about. And I think one place that I think we're missing out on is really trying to look for novelty in terms of their effects on behavior. You can say, and you can combine that with brain imaging data. Like I said, you can look at the brain while it behaves, rather than just laying in the scanner where we don't know what the hell it's behaving and doing, you know. And so, you know, what I really would like to see is some, you know, really new phenomena that come out with psychedelics. And so I give an example to people where, you know, I've said, look, I've learned nothing about the mind from psychedelics. And, you know, I had somebody, Al Garcia Romeo here at Hopkins was like, all right, well, what would satisfy you learning about the mind from these drugs? So I was like, all right, let's pretend somebody was standing on their head on LSD and they consistently saw the color red as green. I was like, that would tell us something about sensory integration that we didn't know about before, that proprioception and visual information is somehow integrated in a way that's imperfect. And it tells us that this algorithm, yeah, it's imperfect, and it has ways in which it can potentially fail. So that's something. I mean, we found, you know, this one effect where we sort of see that essentially this one memory process is impaired and another one might be enhanced. And that's also shown in non-drug studies, and that's associated with things like deja vu and presque vu. So essentially feeling like you're having an insight, even though you don't actually have an insight. Those are things that we know exist in psychedelics. Sometimes people will say deja vu, they sometimes say they have an insight. Clearly not the vast majority of people doing psychedelics don't become, you know, Nobel Prize winning physicists or cognitive psychologists. They're not, their insights about the mind aren't probably actually all that veridical, but they feel like they have it. Did we really even need psychedelics for that? So now I'm providing more information towards this idea that this one memory process down, the other one up, results in these weird phenomena like presque vu and deja vu. But that was already shown without drugs. That was shown with pure behavior. And so, I mean, I do think it's a starting point though. And, you know, it's hard to evoke things like presque vu and deja vu. And so those might be important things to try with psychedelics to kind of further probe. Or like, for example, I was trying to talk about this, the color constancy, the fact that we see everything as one color, even though that it's not, and there's ways in which color constancy can fail. There might be a way, and it seems to fail pretty harshly under psychedelics. That might be an interesting place to see, you know, start looking at with psychedelics. And it's unfortunate that, you know, vision perception researchers haven't broken into this field yet really all that well. And so, I mean, because I think that, you know, just like I think they realize, a lot of cognitive neuroscientists realize drugs don't a lot of times tell us about the mind. They're interested in understanding the mind and the brain. Drugs don't always really tell us how the mind works. We learn about the drugs by using proper paradigms from psychology, but we don't learn about psychology a lot of times with drugs. So I think that's, you know, and I think it's possible. We just haven't
Something
done it
has
yet.
to be happening with the visual field interaction. I think people look past that way too quickly. It's just like, wow, that's trippy. And it's like, no. I mean, when you consider all the just vestibular and visual feed forward and feedback mechanisms, the different structures of the brain that's processing all that, and like what it implies for just threat prediction and pattern recognition alone, I think there's going to be more to the broad spectrum efficacy coming from the visual field than we currently realize. And it'll be interesting when folks like David Olson, who are just trying to work on the compounds that don't have any hallucinogenic effects. I'm just wondering if there'll be certain conditions where we don't see as large an impact. Whereas like, obviously for other conditions, it's probably a good thing to not have all this going on. I know I've just opened a whole new side door here.
Yeah.
But, sorry,
I don't know.
Sorry.
I still wonder if those compounds are actually not going to be hallucinogenic. No one's taken them yet, as far as I know. But yeah, my guess is they'll probably still have some of the effects of psychedelics, especially maybe at higher doses. But I'll be happily proven wrong that when it turns out they're just like an SSRI where they don't, have large effects immediately and they work over time. I think what David Olson's doing is great. So yeah, I guess that's probably where we should maybe end here. But
fair enough.
Yeah,
I know I appreciate you persisting in the beginning to get on here and going through, I don't know what it was. Anyways, thanks everyone for your understanding, patience there. And just once again, my gratitude to you and honestly, all the researchers out there that are doing this, just profound effects in the lives of people like myself, other people I know. I already know about people that are also starting to have very profound effects in terms of chronic pain because of psychedelic research and interactions, and that's REMAP's main focus. But
right. Oh, I guess just real quick, the one comment I wanted to make on that was that the striatum's known for these patterns and for maintaining patterns. And so the one idea is that, especially with things like chronic pain or with tinnitus, for example, is that they might start in the periphery, so outside of like the cerebrum, right? Or at least, maybe at lower levels of the cerebrum, for example, in the case of tinnitus, you get maybe early auditory processing, but they're maintained potentially by the cortex and things that happen in the cortex, or let's say what's happening in the striatum, or how the striatum is perhaps somehow training the cortex, for example. There's kind of this idea that a lot of times subcortical structures train the cortex. And so by somehow breaking that circuitry, you might now be able to kind of undo some of those learned patterns. But I mean, that's about as far as I can take that, in terms of speculation. A little bit like
decreasing a strong prior?
You could say that, except that, okay, how about this?
Please.
Maybe, okay, yeah, sure. Maybe if you're talking about the prior being in the cortex, but if you're saying that it's, let's say, this pattern, this habit formation is in the striatum, that wouldn't go with rebus, because that would be considered a lower level region. Sure. Yeah, I've got, like I said, the whole, I think there could be something there about the priors. I've just got issues about this higher level and lower level thing when it doesn't map onto behavior at all, and their own data don't always map on to, the fact that they get more connectivity from higher level regions to lower level regions, the parahippocampal gyrus to visual cortex. So yeah, I mean, I think there's parts of there, but I think this whole idea of higher level priors, it's just so, it's so high level. It doesn't, I don't know what that speaks to. This is just the idea that people will say. I mean, look, Aldous Huxley said it too. The idea from the concept to the percept is kind of broken. That's just another way of saying that. He said that back in 1950s under mescaline, right? So I just don't know how useful that is. And this is a whole thing about predictive processing that a lot of people kind of wonder, is that like, this is just a theory. It's sometimes a little bit of a theory of everything, that it's unfalsifiable, and it doesn't really, it can just be applied to anything in some ways that I'm not sure where it takes us in terms of predicting novelty. But again, I'll be happily proven wrong. I think there's a whole thing about Americans not caring as much about Carl Friston. I lived in the UK, I should say, for a year and a half. And I probably cared about Carl Friston more at that time. But yeah, I just, want to emphasize, you don't have to go along with what, everybody is saying, that, it's good to think for yourself. And if anything, if I can come back to this idea that like, hey, look, if I keep finding, look, the default mode network, it keeps showing up across all these conditions, behaviors and whatever, default mode network related behaviors are being more affected, or patients with lesions in the default mode network no longer get psychedelic effects or whatever it is, great. I will be happily proven wrong, and I will admit that I was wrong. I hope I do anyways. If I don't, call me out for it if I don't.
rigorous
Yeah.
criticism and examination. I mean, you know, if the worst outcome is that you make it a better model, you know what I mean?
Yeah,
honestly.
And I think that exactly, it's just, this is where people need to be willing to let go of their models. And I mean, I think one thing that, I don't know if he did this just because of, you know, maybe kickback or what, but he did tone down the Freudian stuff in Rebus, and it wasn't fully toned down. And I think that, for example, maybe his priors about Freud have changed a little bit. I don't know that to be true. Maybe they haven't, but yeah, I think that, for example, there is some shifts potentially in the way he's conceptualized, you know, the entropic brain hypothesis, and you know, and same. I don't have a strict model. I'm thinking about something right now, but I'm not going to write about it because I don't want to get scooped, and then two, I don't know if it's real until there's a lot more behavior that I need to see, and that we don't have the funding to do. Somebody give me $1 million, I could find out in a couple of years whether or not this other model is true. But yeah, for the moment, I'd rather hold my tongue than just put out things and try to, you know, connect the dots and read the tea leaves.
I mean, I'd love to see you get the funding for that kind of work. That would be wonderful. I mean, I think multiple approaches create a better. I mean, you know, you're illuminating the sides of a Leviathan in the dark, and like if we're just looking at what's under the beam of one flashlight, it's still hard to get a sense of the full scope of it. So I think what you're doing is great here. If you ever want to come back, we'd love to have you again, and maybe next time we can focus a little bit more specifically around pain. I just felt like we needed to let this like go because there's just so much to cover and introduce right there. So it's like deeply appreciated. Just everyone, thank you for coming. Please visit us at remaptherapeutics.com. You can subscribe there. We'll have this available most likely on YouTube. Also, I say subscribe to our newsletter. You can also, if we recommend taking our course, integrating neuromuscular rehabilitation with psychedelics. If you want to get a sense of like the breadth and depth of what's going on with pain research and psychedelics. We have also, before I forget, in June, either June the 11th or probably June the 25th, we will have the first public Psychedelics and Pain Symposium. So probably about 4.5 hours, multiple panelists, open to the public. Looking forward to that. So Dr. Doss, thank you so much for today. And thank you everybody out there for joining us.
Thanks, man. Thanks for having me.
Thanks a lot.
Peace.
Take care.